Welcome to the Deep Dive, where we cut through the noise and deliver the most important knowledge and insights from the latest research, straight to you.
Glad to be here.
Today, we're plunging into the fascinating world of cancer treatment, with a specific focus on prostate cancer.
Right, it's an area where innovation is moving at, well, an astonishing pace.
Absolutely. And we've got some truly powerful breakthroughs to unpack today.
Definitely some surprising findings.
Our mission today is to distill a stack of recent findings, everything from, cutting-edge clinical trials moving into human patients right now.
To those groundbreaking preclinical discoveries, the stuff that sets the stage for tomorrow's treatments.
Exactly. We're going to give you what you need to know.
And we've pulled from an excellent stack of sources for this deep dive. Primarily recent articles from prostate warriors. They've covered a lot of new drug developments and innovative research.
Good source.
Yeah, and our goal is really to illustrate how these diverse scientific approaches are converging, creating a truly multifaceted attack on cancer.
Okay, so let's unpack this then. We're going to begin by focusing on therapies that are advancing rapidly into human clinical trials.
Bringing hope directly to patients. This is really where innovation moves from the lab bench to the bedside.
Right. Let's start there.
Okay, first one to highlight is BetaBart. It's also known as RV01.
RV01. Got it.
And the big news is it just received investigational new drug clearance, that's IND clearance, from the FDA.
Okay, that's a huge step.
It is. It's monumental. It means a phase 1 human clinical trial is now set to begin probably in the fourth quarter of 2025.
So pretty soon then, what is RV01 exactly?
Well, it's a pioneering radiopharmaceutical monoclonal antibody. Its specific target is the B7H3 immune checkpoint molecule.
Ah, B7H3. We hear about that one. Explain that checkpoint idea for us again.
Sure. For you, an immune checkpoint molecule is basically like a brake on our immune system. Cancer cells often exploit these brakes to avoid being attacked. Now, what's crucial about B7H3 is that it's really overexpressed in many aggressive solid tumors, including prostate cancer, but you barely see it in healthy tissues.
Making it a good target?
An ideal target, yeah. To release those brakes specifically where they're needed most.
Okay, so how does it actually work?
So RV01 is conjugated or linked with lutetium-177.
Now, lutetium-177. That's
Right.
a radioactive isotope, right?
Exactly. This design allows it to deliver highly targeted radiation directly to the cancer cells. The aim is maximizing tumor destruction while, sparing healthy tissues as much as possible.
Now, you mentioned something really interesting earlier. It's clearance pathway. It clears through the liver. Why is that such a big deal compared to, say, kidney clearance?
Ah, yes, that's a really important point.
Yeah.
Many similar radiotherapies clear through the kidneys,
Okay.
and that can sometimes lead to kidney toxicity over time,
Yeah.
due to the radiation exposure.
Right, that makes sense.
The liver, however, is generally more radioresistant. So this hepatic clearance pathway for RV01, well, it might significantly reduce those common side effects like kidney toxicity.
Which could make it safer, especially for repeated doses.
Potentially, yes. That's a safer option. And the preclinical studies back this up. They've already shown very promising results. We're talking significant tumor shrinkage and prolonged survival in the animal models.
Very promising indeed. Okay, building on that idea of targeted delivery, let's talk about another tool, antibody-drug conjugates, or ADCs.
Ah, yes, ADCs.
Often called smart bombs for cancer. And I guess for good reason.
That's a great analogy. Exactly.
Yep.
So next up is BLM-14D1. It's an experimental ADC currently in phase 1 trials.
Phase 1. Oh.
Recruitment's already underway in China, and they're planning international studies too.
And what's its target?
BLM-14D1 targets TROP2. That's a cell surface glycoprotein.
TROP2. And where do you find that?
It's notably overexpressed in various epithelial cancers, but crucially for our discussion today, it's significantly present in neuroendocrine prostate cancer, NEPC.
Right, an EPC, a tough one to treat.
Very tough. So back to the smart bomb idea.
Mm-hmm.
An ADC like this uses an antibody part that homes in on that unique marker, TROP2, which is abundant on the cancer cell.
Okay, so it finds the target.
It finds the target. Then, once it locks on, it delivers a highly potent toxin. They call it a payload. In this case, it's a topoisomerase 1 inhibitor. Delivers it right inside that specific cancer cell.
And the linker technology you mentioned.
Ah, yeah, the linker's clever. It's innovative tech that basically keeps that toxin locked up and inactive until it's precisely where it needs to be, inside the cancer cell.
So it maximizes the anti-tumor effects.
Exactly. and minimizes that collateral damage to healthy tissue. Very precise.
Okay, we've talked about targeted radiation, targeted toxins. What about messing with the cancer cell's own repair mechanisms, disarming it from the inside?
That's another key strategy. And it brings us to SYN608.
SYN608. What is it?
It's a new molecule, currently in a phase 1 study. It's being tested for advanced solid tumors, including prostate cancer.
Okay.
Specifically, it's for patients whose tumors have what are called homologous recombination repair deficiencies, HRR deficiencies.
that relates to DNA repair, right? Like BRCA genes.
Exactly. HRR is a critical DNA repair pathway. Deficiencies here often involve mutations in genes like BRCA, ATM as well. This makes those cancer cells particularly vulnerable if you hit their remaining repair options.
So how does SYN608 do that?
It's quite clever, actually. It targets an enzyme called PARG. PARG.
PRG, not PRB.
No, PARG. A different but related player in DNA repair that these cancer cells rely on heavily. By blocking PARG, SYN608 basically causes DNA damage to pile up inside the cancer cell.
And because the cell already has problems fixing DNA?
It gets overwhelmed and dies.
Yeah.
It selectively kills these vulnerable cancer cells that are already compromised.
So what's really exciting here, potentially, is that this could work even when other repair inhibitors, like PRP inhibitors, have stopped working.
That's the hope. It offers a new angle of attack, potentially overcoming resistance. Plus, initial lab tests suggest it works well on its own and seems to have a decent safety profile so far.
A potential lifeline for some really tough cases, then.
Could be. It's still early days, phase 1, but promising.
Okay, it's fascinating how researchers aren't just looking for single magic bullets anymore, but combining different tools. Building an arsenal, really.
That's a huge trend, combination therapies.
Which brings us to this triple threat approach called LumOnate.
Right, the LumOnate clinical trial. This is a phase 1b study specifically for metastatic castration-resistant prostate cancer, mCRPC.
mCRPC. And it's triple combination.
It is a multi-pronged attack. It combines 3 powerful components.
Okay, break them down first.
First, you have 177 lutetium PSMA INT. That's a radioligand targeting PSMA to deliver radiation, damaging the cancer cell DNA.
Okay, targeted radiation again, component 2.
Second is olaparib. That's a PARP inhibitor, specifically blocking DNA repair.
So hitting the repair mechanism we just talked about. And third
is pembrolizumab. That's an immune checkpoint inhibitor designed to basically take the brakes off the immune system and enhance the body's own T-cell activity against the cancer.
Wow, so radiation, repair blocking, and immune boosting.
Exactly. The synergistic aim is clear. You get direct radiation damage, you stop the cancer fixing itself, and you help the immune system find and kill it. The hope is this combination can overcome tumor resistance more effectively.
Does this build on previous work?
It does, yeah. It builds on findings from studies like Eclipse, Prince, and LuPARP, which look at these components either alone or in pairs. LumOnate takes it a step further.
Fascinating. Okay, we mentioned ADCs earlier. Let's circle back to another one, DS3939A. This one targets a marker found in a lot of prostate cancer cases.
You're right. This is DS3939A, another novel ADC. It's currently in a phase 1/2 clinical trial. They actually started dosing patients back in September 2023 across Asia, North America, and Europe.
Phase 1/2. So, moving along, what's its target?
Its target is tumor-associated MUC1, or you might see it written as TAMUC1.
TAMUC1, and that's common.
Very common. It's a glycoprotein that's overexpressed in about 90% of advanced prostate cancer cases.
Yeah.
Especially common if the cancer is spread to lymph nodes.
90%. That's huge. And what does TAMUC1 do?
Well, it's a key driver of tumor progression and also helps the cancer evade the immune system. So a very appealing target.
Makes sense. And the ADC design?
It uses something called XDA-DC technology. The basic idea is the same. Deliver a potent cytotoxic payload directly to the cancer cells.
Okay.
But what's particularly important here is that the antibody part specifically binds to tumor-specific forms of TAMUC1. These forms are minimally expressed in healthy tissues.
So that ensures even better targeting, more specificity.
That's the goal. Highly targeted delivery, aiming to reduce those off-target side effects.
So this focused approach, targeting such a prevalent marker, could really be a game changer for a large group of patients with aggressive disease. It's all about that precision.
Potentially, yes. It's definitely one to watch.
Okay, let's pivot now. We've covered therapies moving through clinical trials. Now let's dive into some equally fascinating preclinical work.
Right. The stuff pushing the boundaries in the lab, setting the stage for tomorrow's treatments.
Where do we start?
How about with artificial intelligence?
Mm.
There's an AI platform developed by researchers at the Technical University of Denmark and also the Scripps Research Institute.
AI designing cancer drugs.
Essentially, yes. What's truly fascinating here is its ability to dramatically speed up the design of these specialized protein molecules they call Minibinders.
Minibinders. What are they?
They're designed to precisely guide a patient's own T cells to recognize and then kill cancer cells.
Yeah.
Highly specific guidance.
And the AI speeds this up how much?
The speed is incredible. This platform can design effective Minibinders in just 4 to 6 weeks.
Weeks. Compared to what, usually?
Compared to a process that traditionally takes years, it's a massive acceleration.
That is astonishing. And I read something about a virtual safety check.
Yes, that's another key part. The AI screens these Minibinders for any potential harmful cross-reactions with healthy cells before they even hit the lab experiments.
So it weeds out problems early. How significant is that?
Oh, it's a critical leap forward. It massively enhances safety by catching potential issues way earlier. Saves huge amounts of time, resources,
Right.
and the lab tests look good so far. They engineered immune cells called iMPACT cells with these AI-designed Minibinders, and they efficiently eliminated cancer cells in tests.
Wow. And it can be personalized.
That's another big plus. The platform can tailor treatments to unique patient markers. Truly personalized medicine on the horizon. Researchers think this approach might enter human trials within about 5 years.
5 years. Which sounds fast for this kind of complex therapy development.
Incredibly fast, historically speaking.
Okay, shifting gears from A for I. Let's look at a completely different class of drugs. Ones that essentially tag bad proteins for destruction.
Right. Using the cell's own disposal system against itself, in a way.
How does that work?
We're talking about LYA914. It's a novel oral drug being developed for advanced prostate cancer, specifically the castration-resistant form.
Oral, that's convenient, and it's called A.
It belongs to a cutting-edge class called PROTAC. That stands for PROTAC, targeting chimeras.
PROTACs. Okay. What makes LYA914 special compared to older drugs targeting the androgen receptor?
Good question. Its unique advantage is what part of the androgen receptor, or AR, it targets. Unlike older AR degraders, LYA914 targets the highly conserved DNA binding domain of the AR.
Why is that important?
Because it allows it to degrade not just the full-length AR, but also those tricky resistant splice variants, like ARV7.
ARV7. That's a notorious driver of resistance, isn't it?
It is. It often causes therapies like enzalutamide to fail. So LYA914 might be able to overcome that resistance by degrading both the main AR and these problematic variants.
It's basically disarming a key driver that's learned to evade other drugs.
Precisely. And preclinical findings look strong. It inhibited growth of prostate cancer cells, including lines resistant to enzalutamide.
Better than enzalutamide in models.
In animal models, yes. Oral LYA914 showed greater tumor growth inhibition compared to enzalutamide. It really harnesses the cell's own protein disposal system effectively.
Very clever. Okay, next up, something about disrupting cancer's energy supply. Pulling the plug, so to speak.
That's a good way to put it. This involves an unexpected drug combination.
Tell me more.
This comes from a Northwestern medicine study, published in Science Advances just recently, July 2025. They found that combining a MYC inhibitor drug.
MYC, that's a major cancer gene, right?
Huge. MYC is an oncogene involved in, well, over half of human cancers, including prostate cancer. Drives our growth.
Okay, so they used a MYC inhibitor, MYCI-975.
MYCI-975, yes. They combine that with Metformin.
Metformin, the common diabetes drug.
That's the one. An unexpected pairing. Here are the mechanism they found. When you use the MYC inhibitor, the cancer cells try to compensate. They try to boost their energy production by ramping up mitochondrial complex 1 genes.
You fight back, basically.
They try to.
Yeah.
But here's the clever part. Adding metformin blocks that compensatory mechanism. It stops them from boosting their energy supply.
So the MYC inhibitor hits them, and metformin stops them recovering.
Exactly. The cells basically collapse under metabolic stress because they can't generate enough energy to survive the MYC inhibition.
That's fascinating. Does this suggest a way to pick patients who might benefit most?
It does raise that important question. Could doctors potentially select patients by looking at their tumor's mitochondrial gene levels? It hints at a possible biomarker to guide treatment, making it more precise.
A very interesting approach. Now, this next one, It's a fantastic example of thinking beyond what drugs to use and focusing on when. Timing is everything.
It absolutely can be. This is a really significant preclinical breakthrough from the Valdebron Institute of Oncology, VHIO.
VHIO. Okay, what did they find?
So the core finding involves CDK4/6 inhibitors.
CDK4/6, I remember those didn't do so well in prostate cancer trials before.
Exactly. They failed when used alone or even simultaneously with other drugs. But VHIO found that these inhibitors can actually push cancer cells into a dormant state called senescence.
Dormant, like hibernation.
Sort of. The problem is these senescent cells can hang around hidden and then later wake up and cause the cancer to come back. Recurrence.
Ah, so dormancy isn't a cure.
Not necessarily, but here's the crucial part. The study showed that sequencing the therapies is key.
Sequencing, meaning the order you give them in.
Precisely. Give the CDK4/6 inhibitors first, then importantly withdraw them. Stop giving them.
Okay, what happens then?
That withdrawal makes those dormant senescent tumor cells highly sensitive to PARP inhibitors.
The DNA repair blockers again.
Yes. It creates this window of opportunity, a period where the cells are uniquely vulnerable. It's like a 1-2 punch.
So CDK4/6 puts them to sleep, waking them up makes them groggy and vulnerable, then PARP inhibitors deliver the knockout.
That's a great way to visualize it. It significantly improves tumor control in the models because you're exploiting the DNA damage that accumulates right after the CDK4/6 inhibitors are removed. You hit them when they're down.
So this really could be a paradigm shift, emphasizing biological timing and vulnerability over just throwing drugs together simultaneously.
It really underscores how sophisticated treatment strategies are becoming. It's not just what, but when and how.
Okay, finally, let's look at one more cutting-edge radiopharmaceutical showing promise in preclinical studies.
Right, this involves new preclinical data on ATNM-400.
ATNM-400.
It's a non-PSMA targeting antibody radioconjugate, and it delivers actinium-225, a C-225.
Actinium, that's
Yeah.
a powerful alpha emitter, right? And non-PSMA, so it's not targeting the usual suspect.
Correct. A TNM-400 targets a different protein, one that's linked to tumor progression, survival signaling, and crucially, resistance to androgen receptor pathway inhibitors, or ARPIs.
So it hits a mechanism involved in resistance to drugs like enzalutamide or abiraterone.
That's the idea. Hitting a pathway that lets aggressive prostate cancers escape other treatments.
And how did it perform in tests?
What's particularly exciting here is its improved efficacy compared to even established therapies like lutetium-177, PSMA-617, Pluvicto, and also actinium-225, PSMA-617, especially in treatment-resistant models.
Better efficacy. How so?
Well, ATNM-400 required a much lower dose to achieve its effect. That suggests significantly greater potency.
Lower dose, higher potency. That sounds good. Any other standout findings?
Yes, this is truly impressive. When they combined ATNM-400 with enzalutamide.
The ARPI.
Right. In tumor-bearing animals, that combination achieved 40% complete cures.
40% complete cures. In preclinical models, that's remarkable.
It is. Also, PET imaging showed the drug getting into the tumor and staying there for up to 216 hours, but clearing rapidly from normal tissues.
Which suggests potentially fewer side effects, better targeting again.
Exactly. Reduced off-target effects.
Yep.
That combination therapy showing cures is really something.
It really is. Of course, preclinical needs human trials.
Absolutely.
Yeah.
While these results are undeniably promising, it's so important to remember these are preclinical findings. They await that crucial clinical validation to confirm safety and efficacy in humans, but the data certainly lays a very strong foundation for future trials.
Okay, so let's pull this all together. What does this whirlwind tour mean for you listening in? We've covered everything from AI speeding up drug design,
To highly targeted ADCs, new ways to hit DNA repair.
clever combination therapies, sequencing strategies. It's a really dynamic and evolving landscape, isn't it? Rapidly changing how we approach cancer.
It really is. If you connect this to the bigger picture, what's absolutely clear is this relentless pursuit. Scientists are constantly seeking more precise, more effective, and importantly, safer ways to treat cancer.
Yeah.
And these innovations, whether they're just entering those early human trials or still pushing boundaries in the lab, they represent significant leaps forward. Real progress in our ability to fight this disease.
So for you, The learner. Hopefully this deep dive shows just how diverse the thinking is. Scientific minds attacking cancer from, well, every conceivable angle, often using surprising combinations and truly cutting-edge technology.
It's a testament to ingenuity, really.
It is, and as you reflect on all this, maybe consider this question. How will that dramatic acceleration of drug design we talked about, especially with AI,
Yeah.
how will that reshape the whole landscape? Medical research, patient access, maybe even in the next decade?
Right. Will that 5-year timeline to trials we mentioned soon feel like ancient history? Could we see breakthroughs reaching patients faster than ever before?
Something to think about. Thank you for joining us on this deep dive into the future of cancer treatment.
It's been a fascinating discussion.
Stay strong.
And fight on.